fellows, this first question, where does life come from in the first place? That's a pretty big one. How is it that a sort of interested layman can only suddenly realize, you guys had this elaborate, what about this? Did science simply treat it as unimportant or so easy that they'd get to it sooner or later? The question of the origin of life.
Nobody knows the origin of life. Nobody has ever known the origin of life, but people assume, very much like the Babylonians assumed, that it came out of a stinking pond. That's where they say their life came out of, and their gods came out of that same pond. And that's the primordial soup model, and that model persists today. You look in any textbook, from elementary school to advanced college textbooks, and it will speak of the primordial soup model. Life came out of that there were molecules that came together and they formed higher order structures, and that formed a cell, and that led to life. That is a bunch of nonsense.
Okay, hold on. I remember those textbooks with flasks and tubes. So let's take a moment there. First of all, you said something interesting. Darwin's warm little pond. Darwin is working in a great tradition here. The Babylonians had myths involving the primordial soup. So let's take a moment here. The famous Miller-Urey experiments of the 1950s, very simple, 2 flasks, closed system with tubes. One flask has ammonia, methane, and a few other elements to simulate the primordial atmosphere. Another flask has water to simulate the primordial ocean. And then electricity gets introduced to simulate lightning. And what do you know? Amino acids seem to settle out of this. And in early experiments, according to my reading, there were 5 amino acids, which could be described as central building blocks of life, I'm told. In later experiments, they refined it, improved it a bit, and they got up to 22 amino acids. And there it seems to have stopped. The scientists, oh, that's close enough. We can extrapolate from what's going on there. Keep this up for 1 billion years and you'll end up with life. And James Tour says?
It's garbage. That doesn't work. Yes, they made a number of amino acids. They also made amino acids that are totally unnatural. And so they had many amino acids. I think that there may have been a dozen or so of the amino acids, but it doesn't matter. All of them were achiral, which means they didn't have the handedness that one wants. Organic molecules, aside from the very simplest of organic molecules, have two-handedness. You either have a right-handed model or a left-handed model. Your right and your left hand are mirror images of one another. If you put your right hand up to a mirror, it will appear as your left hand. The two are non-superimposable. That's why your right hand can't fit in a left-handed glove. Molecules are like that. Organic molecules are like that as well. He made both of the images. You have to have one and not the other. So there were other experiments that talked about resolving these, separating these. But that's the easy part. That's actually quite easy. But what was presumed from that experiment, which was actually a very nice experiment, I'm not belittling that experiment at all, is that we would very soon figure out the way to life. That's what was assumed. And so that was 75 years ago. So think about that. Think of what has been done in the last 75 years, where you have this whole silicon era, which started in 1960. You have computers, you have space flight, you have landing on the moon, which was in the 1960s, a long time ago. You have all of this internet connectivity, the medical advances. We still don't know how these things come together. You have to take the amino acids and polymerize them, hook them together. That's a big problem. Nobody has solved that.
Jim, may I read a little bit from a paper of yours, one of your 1,000 peer-reviewed papers? I don't understand a word of this, but I'm going to quote a little bit and ask you to help me understand it. Quote, this is the title of the paper: "Are present proposals on chemical evolutionary mechanisms accurately pointing toward first life?" All right. You describe work that you have done in assembling what you call nano cars, tiny little vehicles. Here's a quote. I'm quoting you in this paper. "So here's something to consider as we think about the problem of the origin of life. Designing nano cars, to which you've already devoted about a dozen intricate pages of how you design these tiny little vehicles. Designing nanocars is child's play in comparison to the complex molecular machinery and information processing systems at work in the synthesis of proteins, enzymes, DNA, RNA, and polysaccharides, let alone their assembly into complex, functional, macroscopic systems, close quote. Is this layman correct in saying, as we have learned more about chemistry, as we have achieved the ability to manipulate chemistry more and more accurately, we have learned that the question of how life first began is far bigger and far more complicated and far more elusive than Darwin and Miller and Urey ever conceived?
Yes, this happens all the time. We see this every time we try to make something. The goalpost goes further away. It's not that the cell is evolving. It's that we understand more of the cell. So we're here, we move a little bit closer to maybe solving this. But the cell, the target, has moved miles and miles further away because we're like, oh no, I have to make that too? Oh, I have to solve this problem too? So when you just think a cell is a bunch of protoplasm, the target's not very hard. But then when you learn about the cell and the complexity of it, and you see that biochemistry is extraordinary, and there's so many layers to this that I never even thought about because I didn't have the tools to see it 50 years ago, but now I see it. And then you think about how those might be solved, how you might solve those, and then you see layers of other things you have to solve. So though people may say we're getting closer, the target has moved much further away. So I would never say that it will never be solved. As a scientist, I can't say that. But I just say that the solution is very far from today.
So,
No.
why hasn't the whole scientific establishment said, stop presses,
We're in
fellas,
trouble.
we have a
yeah,
problem?
yeah.
Isn't
That's what I've said. I said time out. I had a whole article called Time Out. I mean, you're sensing the frustration. Welcome to my world. I mean, this is the problem. This is the problem. None of this works. It's a bunch of science fiction. It's really nonsense what these guys are putting forward. And they're building their careers around this thing, and money continues to this day. 40 million euros was just designated to, in 10 years, figure out where life came from. And there's about 40 researchers that in Europe that are going to try to do this. It's really nonsense. We don't even know where to begin. We don't know how to take the simple building blocks. What they're going to end up doing, and I'll make a prediction right here, what they're going to end up doing is something that's analogous to what Craig Venter has already done. They're going to take a living cell and they're going to knock some parts out of it and then put the parts back in that they knocked out and said, hey, look, we made life. You didn't make life. You started with a living cell. I mean, this is what they're going to...
You started with a car, you took the carburetor out of one car and put a carburetor from another car in and said, hey, look, we made a car.
Because they're not going to know where to begin. There's so many problems. It's not just 5 problems. I talk about 5 problems because these are 5 you're going to have to solve, but that's 5 of 5,000 that are facing you that just to begin. None of these do we know how to make the polymers. None of these do we know how to assemble them. We're just lost on every phase. It is a big scam.
One more question. Jim, I'm going to quote you again. I'm quoting your article. Imagine that the world's best synthetic chemists, biochemists, and evolutionary biologists have combined forces to form a team, a dream team. Money is no object. They have at their disposal the most advanced analytical facilities, the complete scientific literature, synthetic and natural coupling agents, and all the reagents their hearts might desire. They have a lot of things I don't even understand, but they have it all.
Have it all.
Carbohydrates, lipids, amino acids, and nucleic acids are stored in their laboratories in a state of 100% purity. Would the dream team, I'm continuing to quote Dr. Tour, would the dream team please assemble a living system? Take your time, folks. Take a few billion years. Nothing? Well. Okay, Jim. This expresses your frustration. I get that. But there's one piece of this that still puzzles me. You say take a few billion years. Isn't that still the argument? That if you give it enough time, somehow or other, it's an immensely complex procedure, but if you give it enough time, enough shocks of lightning, maybe we can't do it in a lab, but the real lab had billions of years at it. What about the element of time?
Time is your enemy.
Enemy?
Time is your enemy. Yes, because those organic
In the 50s, George Wald, famous scientist who worked on the origin of life problem, said that time was the hero of the plot.
Right.
Give it enough time that what's improbable will become inevitable. But Jim has
I'm perfect for you because I was using old textbooks in high school that were probably printed in the
Well, we
50s.
all still
So
get
this
this.
is what's
we
in my
get this.
head.
Yeah, same stuff.
So the compounds decompose. All those beautiful chemicals that they have stored in their labs in 100% purity, after a few years, they're not 100% purity. In fact, the RNA, after a few weeks, has undergone significant degradation. So the whole idea is that, oh, well, somehow one RNA formed and then it took off. If you have one RNA molecule, if you figure the half-life of RNA may be a month in water, you have to change that to a probability. That one molecule has a few hours to survive. It doesn't survive. Just like your pharmaceuticals, it says don't take this after 1 year. Now you want to wait billions of years and see what it's going to become? Everything goes bad with time. These are not even stored in very, very strict conditions, but even if they are, there's always a degradation rate. So time is your enemy. So if you happen to have all of those chemicals, they're all going to go bad. Your time is your enemy. It's the enemy of being able to do these things. In fact, every organic chemist knows this. If they go home for the weekend without treating their compounds properly and bringing them on to a stable state, they come back after the weekend and it's fallen apart. I mean, you pay the price for this sort of thing. So you don't have the time. Time's your
In
enemy.
these Miller-Urey type experiments, there's a problem called interfering cross reactions, where you get, yes, 2 or 3, maybe 4 or 5 protein-forming amino acids, but there's all kinds of other chemical compounds that are also synthesized in those same experiments, and they will quickly react with the things you want and form, in the case of the Miller-Urey experiment, the sludge was called melanoidin, and it's moving in a life-unfriendly direction. So what the chemist has to do is effectively intervene to remove the byproducts that are unwanted to allow only the things that are wanted to continue on in the simulation. But that's
the chemist has to play God, which is cheating.
God, exactly. It's an intelligent intervention, and therefore not a simulation of an undirected chemical evolutionary process.
Let me sum up, if I may, where I think I now understand things stand. I'm going to quote Jim one more time. This is a different article called Clueless on the Origin of Life. Quoting you, 2/3 of a century since the Miller-Urey experiment, origin of life research has not made any progress whatsoever. Not made any progress whatsoever. We've been to the moon, satellites in space, internet, but on the origins of life, zippo. We can even say that suggestions on how life might have formed really show how life probably did not form. Nothing even resembling a synthetic cellular structure has arisen from its independent components, let alone a living cell. Not even close. Are you both willing to stand with that as a good, tight summary statement?
I am, but I have a small difference with Jim on this, perhaps, and that I think that is progress. I think that we've defined how difficult the problem is.
He's playing semantic games.
Yeah, it is a semantic game in a way, but it's an important point because, as I was just saying, that for the chemists to move the molecules in even a modestly life-friendly direction, they invariably, in their simulations, have to remove things they don't want, use purified reagents. They are inputting information into their system, and therefore the logic of these simulation experiments run like this. We're going to do an experiment under conditions that we think model what was on the prebiotic Earth, and then we're going to see what happens. Well, in order for the molecules to move in a life-friendly direction, invariably there has to be an intelligent input into the system to manipulate the chemical conditions to move them in that direction. So what's actually being simulated? Aren't they actually then simulating the need for intelligence to move from simple chemistry to more complex life-relevant chemistry?
But even their intelligence is nonsense. Their intelligence shows that this doesn't work. They're so clueless on this thing. They say, okay, we'll do this and it solves this problem. No, you just introduced 10 more problems. This is a bunch of nonsense. The whole area is a scam.
Okay. Are you down for any of this program, or do you want to just say?
Well, I mean, I wish you could see what I see, because then you would, even if you just take a cell, very simple cell, and you ask scientists to look at this cell, what is going on? If you just put molecules, align them in that way, is it going to run? Is it going to operate? We don't even know what got this cell going. We don't know. I mean, so you should ask a scientist, a cell just died. What is it we just lost? I mean, all the molecules are right there. Could you get it going again? We don't even know how to describe what life really is when it comes to even a simple cell. We don't even know that. That's how clueless we are. I wish you could see this, because you don't understand the magnitude of the human deficiency in even understanding what life is in the simplest of organisms. And people will say, well, life was much simpler at the start of the evolutionary process. This has already been calculated. Bioengineers have already figured out what is the minimal operation that you need. What are the fewest components that you need to have a cell operating?
Right.
And you take that and, okay, so let's say we make all of those components. By the way, none of those 15 components have been made. None of them. Now, if you could make them, now what would you do?
Under realistic prebiotic conditions.
Yeah, even if you could make them in your current lab. I'll give them their own labs. Make them, or just take them from a living cell. Go to a living cell, use that as your supply chain. Take it from the living cells. Now what do you do? Just putting them in proximity? Is this going to run? Nobody in the right mind would say yes. No scientist would say, it's not just putting them together. You don't know how to put them together, but even if you could, now how does it even start running? We don't even know what we're going toward, which gets back to that philosophical experiment that his professor was talking about. What are we going toward in life? We don't even know what life is. Molecules don't care about life. Molecules are random. They don't move toward life. There's no propensity for a molecule to move toward life. It has no brain. It has no action that wants to bring it toward life. They don't know. I don't know what you want me to do. I'm just here. What do I do? And then even if we could say, I want you to move toward life, you'd be like, okay, just tell me which direction to go. I don't know.
Jim has an amazing talk.
I'm going to ask you, because you are such a brilliant man and you understand things at such a level of detail. If I take a petunia seed and it comes to me in the packet from Burpees, and all these seeds are little dead things, and I put it in a pot and I put some water on, now I can predict, and you can predict, that we'll come back in 2 days and there will be a little shoot, and a week later there will be a flower. But if I say, why? How did that happen? Are you telling me that at the moment of life, at the moment something passes from inertness to life, that remains a mystery to us today?
Yes, it remains a mystery. What is the very even thing we're describing when we talk about life? I've had scientists, I used to do this demonstration for my own children. I said, watch, I'm having scientists over tonight. Watch what's going to happen. I said, you have a cell, it just died. What is it you just lost? And then they start arguing with one another. There were 2 questions. I said, what is it you lost, and how would you get it going again? They could never get to how we get it going again, because they could never figure out what it is we just lost when the cell died. That's how mysterious life is. Now, that's not to say that we won't one day know. I can never say that. As a scientist, I can't say we will never know. There's a whole lot of things we've figured out. If you asked a man in 1700, will we ever be able to walk on the moon and come back? He'd be like, no, we don't even have space flight. I mean, how can I say? How can I say? I don't know what the future holds, but all I know is it's not going to happen tomorrow. It's not going to happen in 10 years with 40 million euros being thrown at this thing. That's more money down the drain. You're better off letting the taxpayers keep that money, because they're going in the wrong direction. We don't even know what we're going toward. Okay. What I'm doing is I'm pointing out the obvious to everybody. There are so many scientists that don't work in the area of origin of life that agree with me. They see exactly what I see. This is not unique, this is not hard to see. Everybody sees what I see, except these guys that are working in the field. That certainly scientists are seeing this, but they're not speaking up. They're not speaking up because they're afraid to, but they're seeing the barriers that are there, and there is no solution. The only ones that are not speaking up are the ones that are making their living from this thing. But nobody wants to speak up and get this abuse. My hope is that as young people come into this, these organizations will begin to change. That's my hope, that people will begin to be more open-minded here.
Okay.
There was a major article in Nature just a month or so ago, Jim, right? By two authors that are leading origin of life researchers, Johanna Xavier and Nick Lane. And Johanna had previously actually commended both Jim and me for raising these serious problems. And then she went on with Nick to write an article in Nature saying, hey, we can't keep sweeping this stuff under the rug. And so I think there is a shift starting. I think a lot of origin of life researchers are pretty terrified of Jim Tour because he knows the chemistry as well or better than they do. And he's saying, he's performing this chemical accounting and saying that these experiments are not plausible. They're not getting results that are plausible. They're not consistent with what we know about what would have been happening on the early Earth. We need another approach.